Genetics of cystic fibrosis. So the genetics of cystic fibrosis, this is a common disorder occurring in 1 in 3,500 individuals of Caucasian descent. The carrier frequency in the Caucasian population is 1 in 25. That means if you look at 25 individuals of Caucasian descent, one is actually a carrier for one non-functional copy of the CFTR gene. Now, the frequency in the Asian population is far less common, almost 10 times less common, which is 1 in 31,000. This is just a pedigree of cystic fibrosis, or really any autosomal recessive disorder. The full filled box means that the individual is affected, and you'll see there are a lot of carriers out there, but carriers in general do not have symptoms before you see an individual that's affected. There are some hypotheses why this is so common in the Caucasian population, and one is that being a carrier for one non-functional copy of the CFTR gene gave selected advantage during the bubonic plague, or that it was a GI kind of problem, and that they survived this, and therefore if they survived being a carrier of one non-functional copy of this chloride channel, that they were more likely to pass on their genes. So there were over 1,300 changes in the CFTR gene, 23 common mutations representing about 88% of Caucasians, 69% of African Americans, and 57% of Hispanics. And screening for the presence of being a carrier, or carrier screening, is an inexpensive method that can look for common mutations. Sanger sequencing is a method that's a more expensive method. So one of the methods that's kind of a combination of PCR is something called ASO, or allele-specific oligonucleotides. So you do PCR for common mutations. So, for instance, we're looking at common mutations. So the delta F508 is one of the most common. At site 308, there's a deletion of 3 base pairs. So instead of it going CA, CC, AA, GA, it goes C, A, C, C, A. And then this is all missing. So then it goes to A, T, G, A. And this changes the amino acid sequence. So right in the middle, there's a deletion of 3 base pairs. So the ASO method is a method in which we have we amplify either the normal allele or the delta F508. And so if somebody has both DNA for the normal and the delta F508, that makes them a carrier. If all they have is the delta F508, then they're affected with cystic fibrosis. If all they have is normal, then they have normal DNA or carriers. So these individuals were both carriers for one copy of the normal CF gene and one with this deletion of the delta F508. In this child, both non-functional copies of the gene were donated. In this child, both normal copies were donated. In this child, one parent donated a non-functional delta F508 and the other donated normal allele. Neither one of these individuals, both of the either carrier or normal alleles, have symptoms of cystic fibrosis. So let's look at carrier testing. And again, carrier testing is detection, right, to determine who in the population carries this. So because the common mutations are present in the Caucasian population, the carrier the detection rate of looking at the CFTR mutation is actually pretty high, 90%. So, as I said earlier, 1 in 25 individuals of Caucasian descent are a carrier of CF mutation. But if you pick up 90%, after the testing is negative, you got down to 1 in 227, sorry. Individuals of Ashkenazi Jewish descent, which is a subset of European individuals, 94% mutation rate is identified. And there are 3 super common mutations, one being the delta F508. Before carrier frequency, their risk is a little bit higher than the Caucasian population at 1 in 25. However, after completing genetic testing with a 94% carrier frequency, the risk is 1 in 384. So if you're Ashkenazi Jewish and you have negative carrier testing, your chance of being a carrier for CF is incredibly low. Let's look at the African American population. Their risk is a third almost of that of the Caucasian population, but we only pick up 65% of mutations. So the residual risk after testing only goes down from 1 in 61 to 1 in 175. Hispanics, a little bit better. They're a little higher, but a little bit better in detection. But now, even though the risk is more than twice as much before testing, it's pretty much the same as Caucasians after testing. And Asians, even though the risk is pretty low, 1 in 94, we only pick up a little bit over 50%. So it only drops to 1 in 205. So the truth is, after testing, somebody, even though they have the lowest risk before testing, they really haven't lowered their risk very much. And the reason I bring this up, and this is true of a lot of different things that we do carrier testing for, what is the detection rate? Because if it's 94%, then we're pretty, you really lower your risk of still carrying this mutation after we're done looking for common mutations. But if they're only picking up 55%, then you're really not lowering it that much. So cystic fibrosis is caused by a gene located on chromosome number 7 called the CFTR gene. And it codes for a chloride channel. And when not functioning correctly, it causes thick mucus membrane, thick mucus, which blocks airways, and it also gets stuck in the pancreas. So respiratory. This is the major cause of morbidity and mortality because of these viscous secretions. GI, meconium ileus. 15 to 20% of affected babies will have a meconium ileus. And in fact, if you have a meconium ileus, there's about a 75% chance that you have cystic fibrosis. So it's definitely something to think about. Malabsorption, because of the GI issues, can result in failure to thrive. Pancreatic insufficiency. And then in males, bilateral absence of the vas deferens. And we're going to talk about some of the milder forms of CF, that could actually be the only symptom. When the chloride channel, so this chloride channel is in the cell membrane and it transports chloride. And when it's not doing that, it results in this thick mucus. So this is just a little diagram showing this is what it should be doing, and when it's not working right, we're not getting, you can see it's not going through the way it should. So as I said earlier, there's over 1,300 mutations, but this delta F508, this deletion of 3 base pairs, is the most common, resulting in 70% of cases have at least one delta F508 mutation. So again, instead of isoleucine, phenylalanine, glycine, valine, we get isoleucine, and then we get glycine, valine. Because we've lost these 3 base pairs, we don't have that phenylalanine. Now, the other thing I want to talk about is different types of gene mutations. So, a nonsense mutation, you get no protein. So, you transcribe your DNA. What should happen is you transcribe your DNA. It results in the transcript goes to the Golgi. Gets transcribed on the ribosomes. It goes to the Golgi, and then you end up with the Golgi brings these proteins up to the cell membrane, and you transport chloride. A nonsense mutation, a premature stop codon, results in no transcript of the DNA. Therefore, nothing goes to the ribosome, to the endoplasmic reticulum, none ends up in the Golgi, and you get no CFTR proteins in the cell membrane. But there are a lot of other things that can occur as well. And one is a problem with trafficking. And a trafficking problem is the protein is transcribed, it gets into the endoplasmic reticulum, but when it tries to go to the Golgi, it doesn't fold correctly, so the Golgi just gets rid of it and nothing gets up here. Or it gets up to the cell membrane, but it doesn't, it's another missense mutation. It gets into the endoplasmic reticulum, the protein's made, the Golgi brings it up here, but it doesn't transport chloride. Or it doesn't transport efficiently. Or we don't have as much, or it gets broken down faster. These are all the things that can go wrong with these different type of mutations. So personalized medical management. There are a number of medications that are being developed to address the specific problem in the protein function resulting from the change in the gene structure. Splicing defects are when we have no protein getting onto the surface, therefore we can give one mutation if you have a nonsense mutation. Trafficking defects, when we're not able to fold the protein correctly in the Golgi, we can give different medications to get into the Golgi to help kind of shove it into space. And gating defects, we can also, there are specific medications that can help that protein work better. So as I said earlier, there are a number of diseases associated with the CFTR mutation, including cystic fibrosis, which is the classic, but also just bilateral absence of the vas deferens can be milder mutations within the CFTR gene, that the only symptom is in male infertility. And hereditary pancreatitis can also be found with 2 mutations of the CFTR gene, but doesn't result in the classic lung disease, but still 2 mutations in the CFTR gene. Hereditary pancreatitis, recurrent severe pancreatitis. So the delta F508 mutation in the CF gene is defined as a truncating mutation, a splice site mutation, a narrow channel mutation, post-translational defect of the folding, or gating defect. So let's go back here, and where's our delta F508? It's right here. This is a problem with the folding of the protein. And that concludes this lecture.